So welcome everybody to another AFREN webinar for the last one for this year. Today I am going to start by introducing you to Dr. Lisle Snyman. Oh, sorry, I should start first of all with a few housekeeping questions. At the bottom of your screen is the Q&A button. If you have any questions during this seminar, please write them in the Q&A section so that we can get back to you on those questions at the end of our seminar. We have quite a few participants, so it's good to be able to do those at the end. Also, AFREN would like to ask you if you might be able to do a survey at the end of this seminar also. This gives us a chance to make sure that the information that we're providing you is there to help the growers and advisors to stay informed about fungicide resistance and what are the kind of things that you want us to tell you. So please do that survey for us at the end. So without further ado, let's start again. I'd like to introduce you to Dr. Lisle Snyman. She is the Principal Plant Pathologist with the Department of Primary Industries in Queensland. She's been with the department for 10 years, based in Warwick, and she leads the Winter Cereal Pathology Program. Their research and diagnostic service activities mostly focus on foliar diseases of barley and wheat, and today she's going to be talking to you about barley diseases and management. Over to you, Lisle.
Thanks, Nola. Welcome everyone, and thanks for joining us today. As Nola mentioned, I'll be talking about barley diseases, specifically foliar diseases, and appropriate management strategies. So we all do know that the environment influences disease. This will determine, what diseases we get in a given season, when these diseases will start to develop, and also, will it be low infection or will it be severe infection for the season? So this particular graph just indicates the rainfall for Warwick for the years 2022, 2025. So that's the monthly rainfall. And that is the station that we do conduct most of our research at. And you will see that for 2025, we had quite a wet season, and that also comes on the back of, a really wet period at the end of 2024. The only months that rainfall were quite below the long-term mean was in February and then again in August. In general, for Warwick, August and September are quite dry months, but it was still particularly dry for August with the forecast of quite a wet spring. So currently, the Bureau of Meteorology, they still forecast a wet spring and early summer. So with that in mind, we can expect probably a little bit of an increase in disease if that is going to be the case. So for us in Queensland and, more the Downs, the Southern Downs, Darling Downs, so what are the barley diseases that we currently focus on and we regard as quite important? That's generally mostly leaf rust of barley, and then the two net blotch diseases, net form net blotch and spot form net blotch. I have decided not to talk about powdery mildew today, and for a couple of reasons. Firstly, powdery mildew in Queensland is usually quite a problem early in the season, but then as it starts warming up and becoming drier, it tends to taper off. Second reason is that there's quite a big shift towards European varieties, RGT Planet, Neo, CL, or Neo and Xena, and there's quite a few others. And these varieties carry the MLO resistance gene, so they have really high levels of resistance to powdery mildew, and that's why the focus will be more on the other diseases. So in terms of leaf rust is quite common in in the growing regions of Australia, and in fact all over the world, and in everywhere barley is grown, leaf rust can be a problem. It can have a significant impact on yield. We've seen losses of more than 60% in susceptible varieties, and we do know that it's got quite a big impact on quality as well. Leaf rust spread by airborne spores. So these can travel long distances and they can actually result in quite rapid disease spread across large areas. The green breech is specifically very important for the rust diseases. They aid your rust pathogen. They are biotroph pathogens, so they do need a living host for survival. And the green breech aid them to survive over summer. And then, if conditions are favorable early in the season, you can have quite high inoculum loads that can really then attack your crops at a very early stage. We do know that the more susceptible the variety is, the higher the disease level it can carry, and then that results in a higher yield loss. If we do have large areas grown under susceptible varieties, you can have really high inoculum loads, and this put pressure on the pathogen. They mutate and they acquire virulence for genes that currently we can use for resistance in varieties. In particular, for the leaf rusts, there has been some work done and resistance to several of the DMIs have been identified, and that's across all of the growing regions of Australia. It's not limited to a specific region. For the net form net blotches, these two diseases, they occur in most seasons. We can't say which one of those will be a bigger problem in a specific season. And it depends on a lot of factors, definitely environment, but then also, the level of susceptibility of varieties that are grown at large in these areas. Due to a few reasons, it's quite important that you are able to distinguish between these two diseases. Early in the season, both of them only starts as tiny little brownish, blackish spots, and we can't say which one it is. But then as the season progresses and the symptoms, get bigger and more of them, we can then tell the difference. Spot form net blotch is definitely, it is a spot. So they are kind of really dark color, almost like a blackish color. They are circular and they have that yellow chlorotic halo or just a yellow ring around them. As the season progresses, they can get quite long and they can cover quite a big proportion of the leaf in susceptible varieties. In terms of net form net blotch, they're definitely quite lighter, more like a brownish color, light brown color, and they really resemble like little ladders on the leaf. Both of these diseases can cause yield losses in excess of 40, 45 to 50%, and they also have quite a big impact on grain quality. Another difference between these two diseases is that both of them are stubble-borne, but net form net blotch is also seed-borne, and because of that, it has an impact on a little bit of different control measures. And it also impacts a little bit different on the spread of the disease because it can easily carry over from one season to the next in seed. So GRDC currently have a net form net blotch consortium of projects, and we're involved in a few of these. One of them is Program 1, the monitoring of net blotch populations for new virulences of industry significance. This project is managed by University of Adelaide, SARDI, and the aim of this is to equip growers and breeders to respond to new virulent net blotch pathotypes that are emerging nationally. So every year we receive leaf samples for the net blotches, which is done pathotype on a set of about 30 genotypes, and we use that to distinguish between the isolates. It is done for both of the types of the net blotches. So Agriculture Victoria does the pathotyping of the spot form net blotch, and we do the pathotyping of net form net blotch here in Queensland. And if you just look at the numbers, and you will see that 2024, we received 111 samples compared to kind of roughly around 60 samples in previous years, so 111 in 2024. We have just finished the pathotyping of that recently, so don't necessarily have all of the final results yet. So there are three isolates that are common across Australia. They are NB85, 73, and 350. And we've got data going back for much longer than 2020, but this is just kind of the recent scenario. And if you have a look at the NB73, the blue one there at the bottom, the component of the total number of isolates that we pathotyped in each year, you will see that that has increased, from 12% in 2020 to 2023, it was 63%. And we do expect that that would even, be higher once we get the final analyzed results of 2024. And if you look at this, you will see that there was a clear shift in the isolates that we collected across the years in terms of pathotyping. So NB73, if you look at 2020 there, you will see that, it was definitely present in Queensland in 2020. That's the one that we kind of refer to as the old shepherd pathotype. So it has always been present in Queensland. But if you look across the years, you will see that there was, just an increase in that blue, a little bit more every year. And we can attribute that to the presence of a lot of these European varieties that we currently have in our system, and they are susceptible to that specific isolate. And as I mentioned, we do think that that is going to increase even more. So yeah, I mentioned we do the pathotyping. So please, we can't do that without collaborators. So we ask growers, agronomists, breeders, if you do come across net form net blotch in your paddocks, in your trials, please send us some samples. Not necessarily 20 samples from the same location, but we do appreciate some samples. And if you do send them to us, please, as much information as you can send with it. Location is really valued, and then of course variety, as we do know that this impacts on the isolates that are present. You can send these either by mail or hand deliver, whatever's possible for you, to either Hermitage Research Facility in Warwick or Lisle in Toowoomba. You can mark it for my attention. Really, if you have something that you want to get to us and you just can't, give me a call, let me know. And DPI have a lot of staff that works out in the regions, and we would be able to arrange something to get those samples to us. Please, where possible, if you can send it to us, or as soon as possible. It's not ideal to drive around with them on the dash of your ute for weeks. They do go crisp, and then we can't do much with them. Please don't send them in plastic. The smallest amount of moisture will make them go moldy, and it just result in a whole heap of saprophytes growing on them. Also remember that it is not only net form net blotch and spot form net blotch samples that we're asking. You're welcome to send us any queries for diagnostics. Sometimes we do know, especially in barley, we get a lot of spotting in barley, and a lot of it is physiological or nutrient deficiencies, and it is not disease. And we really do want to make sure that there's not unintended fungicide use for something that is not regarded as a disease. We do ask that in terms of rust samples, please don't send that to us, send that directly to the Plant Breeding Institute at the University of Sydney. They do have on their website, a guide on how to send those samples, what they require. And from my understanding, I think they're also working with GRDC on publication of a fact sheet on, how to submit samples to them. If you send it to us, we will just forward them on to PBI. Another project in that Net Farm Net Blotch consortium is the integrated management strategies for Net Farm Net Blotch in low, medium and high rainfall zones, and we do manage that project here in Queensland. We do know that Net Farm Net Blotch have an impact on yield and quality, and it decreased grower profitability as a result. But there's still so much that we don't know, and we do really want to understand that interaction better, and also, understand the impact of the costs involved in managing these diseases. We also do know that stubble retention as a practice have an impact on the presence and the level of disease because both of these diseases are stubble-borne. So the aim of this project is to develop integrated disease management strategies and determine the cost benefit of implementing these in each of the rainfall zones, and then also deliver an extension strategy that ideally will be adopted by growers. We do know that management practices varies big across the rainfall regions. And we also do know that, it's really hard for us to really fully control these diseases without the use of fungicides. But there are quite a few rules when it comes to fungicide use, and we do want to make sure that it is cost effective and we don't apply fungicides without getting the return on our investment. And we also have to ensure that we sustain the efficacy of our fungicides for the longer term. So if we look at these trials, we have collaborators here in Queensland and then also in New South Wales, Victoria and SA, and trials are conducted in all of these different rainfall regions. We do know that, as I mentioned before, practices are quite different depending on environment and, of course, on the rainfall zone. And therefore, there's quite a big difference in how these trials are conducted, because we do want to look at local practices and what growers do in the region. So therefore, there's a big difference in terms of varieties that are used in these trials, but we do want to try and get an idea of the impact on varieties with different resistance ratings. We also look at plus or minus seed treatment and then also different mode of action. The same with foliar fungicides, looking at different mode of action of foliar fungicides, and then of course different times of application, since it varies in the different regions. We also look at the presence and absence of stubble, and then also specifically at the presence of infected stubble. The University of Southern Queensland is also involved in this project, and one of the big components that they are contributing to is that they do all of the genetics, and they look at the changes in the pathogen population over time in these environments. So I'm not going to dwell on the AFREN principles. I'm pretty sure, a lot of you have seen this over and over again. And in future, I suppose we will put a lot more emphasis on these in terms of, using integrated disease management strategies, and not only rely on the fungicides that are there kind of at the top of the triangle. I will discuss some of these a bit more in detail on this slide. And the first thing we want to emphasize is variety choice. There's a lot of information available on the NBT website, and the ratings are available for the different states. And it's important, we would really want to ask growers where you can, use the variety that's best suited to your environment, but also have some level of resistance. In an ideal world, we would have varieties that are resistant to all of the diseases, but we know that that's never going to be achievable. So what we do ask is look at, what's your situation around your farm, what are the disease risks, and then choose your variety accordingly. Look at something that at least have an MS or moderately susceptible rating to those diseases that are of concern in your area. And then, you use that variety choice basically as the start of all of your other management strategies. Crop rotation is very important. The best is for the net blotches specifically to look at something that's not a host. So look at, your pulses, chickpeas, mung beans, things like sorghum or maize, whatever fits in your rotation strategy. Stubble management, I mentioned both of these diseases are stubble-borne, so they will be carried over in the stubble from one year to the next. And also remember that stubble breakdown, it depends on the environment, how quick that stubble breakdown. So it's not necessarily only last year's or last season's stubble that can cause a problem, especially if it's dry. It can take quite a few seasons for that stubble to break down. We do want to look also at time of sowing. We do know that because of softer conditions earlier in the season, that early sown crops are often a bit more vulnerable to disease, and then they also create then kind of like an environment with higher disease levels for the crops that are sown a little bit later. In terms of the green breach, that is specifically important for the rust diseases. I did mention they are biotrophs. They do need that living host for survival. So if you provide them with a green breach over summer, it makes it possible for them to be early in the season present and often at quite high levels, and you can have high levels of disease very early in your growing season. The other thing is, I'll maybe just go back to stubble management for a second, is also remember if for some reason we really do not recommend that, but I do know that, there are people and for various reasons that sometimes go back into stubble with a barley crop. If you really have to do that, make sure that you do not plant the same variety than what you had in that crop last year. And also ideally plant a variety with a higher level of resistance than what you had in last year. So in terms of monitoring of crops, we do ask that growers do not only rely on their agronomist to monitor their crops for diseases. It is agronomists are like everyone else, they're time poor. They don't have the time to really walk through your crop and spend hours in it looking for disease. And as a grower, that's your responsibility to go through those crops and make sure that you are aware of what diseases are present and, what the levels they are at. So the last one is fungicides is, as I mentioned before, it's not always possible to control these diseases without some reliance on fungicides. However, remember that fungicides, their job is to control disease. So if there's no disease, there's no need for you to apply fungicide because you will not gain anything by that. The other thing is also don't wait until you have epidemic disease levels before you apply fungicides. We do know that their efficacy is not the same at epidemic levels than earlier in disease onset. And that also then provide an opportunity for some of these virulent isolates to survive the fungicide application and, then build up resistance against the fungicides. Another one is seed treatment, net from net blotch I mentioned before. It is they are carried over in the seed. So it is quite important that you do use an appropriate seed treatment. However, if you had high levels of net from net blotch in your crop last year, we do recommend that you rather get some new planting seed for the season and don't use those seed with the high infection levels. That is also important for something like smut. And also maybe I just want to say with variety choice, we have seen with the data that I have shown that variety choice has a massive impact on the presence of diseases and also on the presence of particular isolates. And we have seen that with, you know, an increased uptake in the European varieties, specifically for net blotch. I do maybe just want to say that these management strategies, even though the focus for this is net blotch, most of these is, you know, usable for all of your foliar diseases. Some of them, like managing the green breach, more relating to your rust diseases. And lastly, I just want to say that remember that our fungicides are really important for us, and we do want to make sure that we look after them so that we can, you know, make sure that they are effective for us for use in the future. So thank you for joining us, and my contact details are there on the screen. Like I said, please send us some of those samples, and also you are welcome to contact us if you have any diagnostic questions for you know, for your purpose. And thank you so much.
Lisle, that was really informative. So now I'm going to move on and start by introducing myself. Sorry, I didn't do that at the beginning. My name's Nola de Souza. I'm the Australian Fungicide Resistance Extension Network Coordinator. And AFREN is a GRDC initiative that consists of a nationwide network of plant pathologists, fungicide resistance experts, and communication and extension specialists. And we have the aim of helping growers and advisors to stay informed about the prevalence and management of fungicide resistance in their region. Now, just make sure that I can go forward here. So I'm just going to get our communicators to give me control over the screen. There we go. Thank you, Bridget. Yes, sorry. So we want to keep you informed about the prevalence and management of fungicide resistance in your region. So today I'll be giving a general update of Fungicide Resistance 101. And first of all, let's start with, I don't think I've got controls. Okay, here we go. So what is fungicide resistance? So the definition of fungicide resistance is when a previously effective fungicide fails to control that disease in the field, even at the maximum recommended rate and despite being applied correctly. But there are many things that can result in fungicide failure that are not necessarily fungicide resistance. Some examples of these might include application in windy conditions, which results in a poor spray coverage, or incorrect sprayer calibration. But another big reason is the incorrect diagnosis of disease. So is it really disease? I have borrowed this slide from one of our AFREN network members, Steve Simpfendorfer from the Department of Primary Industries and Regional Development in New South Wales, where he gave a map of all of the areas where he was sent disease samples to be diagnosed. And what he found, I'm just trying to get control of this, is that the top 3 had nothing to do with disease. So people were sending in samples of disease where environmental, insect chewing and nutrient deficiency were some of the things that were not related to disease at all. And that resulted in, sorry, this has got a really bad lag, 68% of samples not being related to disease at all. Stephen Simpfendorfer gave us a really excellent webinar that I'd like to let you know is on our AFREN website. So if you'd like to go and take a look at that, please do. It's really informative and will help you to understand a bit more about disease diagnosis in your paddock. So is it really disease? Something like frost damage or like herbicide drift will not be fixed by using fungicides, because fungicides will not fix anything except fungal diseases. So if you can get a correct diagnosis from the beginning, this will prevent unnecessary fungicide application and reduce fungicide pressure, which is one of the things that drives fungicide resistance. Early diagnosis of that disease and the decision to spray also reduces disease pressure, which is another thing that drives fungicide resistance. So is it really fungicide resistance? Once you know it is a disease and you've decided to spray, you can know that the fungicide, then you need to know that the fungicide has been applied correctly and that it's clearly linked with an unacceptable level of disease control in your paddock. And once you have noticed that loss of disease control, then you can call your local state pathologist, or you can call your agronomist, and they can send samples in to be confirmed in the laboratory. So how does sorry, I'm really distracted by this lag. How does fungicide resistance happen? So fungicide resistance occurs when there is fungicide selection pressure. So what you can see on the left here is a naturally occurring resistant fungus to a fungicide within a population of sensitive fungicide. When they cause disease and the grower comes along and applies that fungicide, and those few individuals that survive can reproduce over time, and that includes the naturally occurring resistant fungus. So by applying that same fungicide with the same active ingredient enables that resistant population to survive and multiply over time. And if we look at this in an idealized graph here, if you're applying a group 3 fungicide naturally to a sensitive population over and over again, it will keep that under control. But if you have a fungicide resistant population individual, then that population is not controlled by that same application. And that's when you will find fungicide resistance. So contributing factors to fungicide resistance are that overuse of the same fungicide, which drives that fungicide selection pressure. Also, poor integrated disease management, a lack of genetic resistance within the crops, and a pathogen-conducive season, which all combined result in driving that disease pressure. And what we have to understand is that fungicide resistance is just a numbers game. So if you have an increase in disease pressure where there are resistant individuals that are under fungicide selection pressure, then fungicide resistance will occur. So why do I need to care? If it's just naturally occurring, why don't we just wait and see what happens to us? Well, let me tell you about the barley powdery mildew outbreak in just over a decade ago. What you can see here in this graph is a depiction of what happened over that season, where back in 2008, what we had was a high percentage of barley area that was growing very susceptible varieties of barley. And back then, the use of fungicide was quite prevalent because it was cheap and it was easy, and it could easily overcome the barley. It could easily control the barley powdery mildew. But then that started to change. So in 2008, what we had was 1.4 million hectares of infected barley crops, and they were all producing spores. And it turns out there was some resistance within those spores that started to spread. Over the next 3 years, there was a $65 hectare loss to barley powdery mildew, which resulted in a $300 million cost to for the biggest fungicide outbreak that was ever recorded, and that was in WA. And that's when everybody had to start scrambling to change those barley varieties into a much more resistant. And since then, fungicide resistance across Australia has become evident in a lot of different diseases against a lot of different fungicide groups. So because Lisle was talking about net blotches, I'll tell you what is new in the net blotch fungicide resistance at the moment. And if we drill down a little bit further into that table that we saw there, what you'll see here is all of the open circles indicate a lab detection only. So we have found in the laboratory that there are mutations associated with resistance or reduced sensitivity to these particular fungicides, 3, 7, or 11. And these are a big warning sign that fungicide resistance can develop under high disease pressure and that fungicide selection pressure, because these naturally occurring individuals are present there in the field. New South Wales, you can see, is faring the best with mostly lab detections, but there is some group 3 resistance that has been noticed in the paddock. And WA is currently faring the worst with all of these fungicide group resistances reported to be impacting fungicides in the paddock at some point there. I want to draw your attention to the combination of fungicide resistances of 3, 7 and 11 in WA and South Australia. Tell you about triple mutant fungicide resistance that we reported this year. So what we found was that there was resistance to these 3 fungicides groups, the only ones registered for net blotch, that were present in single isolates, which can be really concerning. We found those in WA and we found those in South Australia. And we also found one isolate in Victoria. However, the Victorian isolate wasn't actually associated with any loss of fungicide effectiveness in the field. So this was the case study that happened in WA, where you can see in two different paddocks. One was growing Planet, one was growing Laperouse. We found these triple mutant net form net blotch isolates in both of these paddocks. Planet has a susceptible to very susceptible rating, whereas Laperouse has a higher rating of just moderately susceptible. The crop rotations were the same: canola, barley, pasture, canola, barley, and both of them were grazed. There wasn't a lot of difference in fungicide applications. They both got a seedling treatment. They both got a seed treatment upon sowing, and the first and second foliar fungicide applications were the same. But they found in the Laperouse paddock that the third foliar fungicide was not required. And this resulted in the RGT Planet paddock, which was severely susceptible to that, then the disease was not controlled. But in the Laperouse paddock, there was a fairly clean paddock there, and they did get control. And what we can garner from that is that with Laperouse having a higher susceptibility rating or a better resistance rating against net form net blotch, resulted in helping out, keeping that disease pressure down so that they could control it. And this is what it looked like in the paddock, where Planet there on the left was severely infected by net form net blotch, and Laperouse was looking nice and clean. So what can we do about this? So the AFREN principles, and I'd like to reiterate what Lisle was saying, is that variety selection should be the foundation of what you choose. So you need to avoid those susceptible crop varieties, as you saw in that previous picture. That made a huge difference against those triple mutant NFNB isolates. You need to back this up with rotating your crops. Use time and use distance to reduce that disease carryover. Don't plant barley on barley where you can where it puts that barley into a disease situation where there's already a high disease pressure from the stubble that's there. And then only spray if it's necessary and apply those fungicides strategically. So we recommend that you rotate and you mix your fungicide groups. So that means that we're going from this idealised graph previously, where we have our fungicide resistance population increasing in the paddock by those continual group 3 fungicide applications, to something like this, where if you mix those group 3s with other fungicide groups, that you can still keep those resistant populations under control. Now, this is the case for populations where you have just individual resistances, but with those triple mutants, you may not necessarily get that control. So you really do need to concentrate on applying the fungicide resistance 5 principles and starting with those, avoiding those susceptible crop varieties and rotating them. Always rotate your crops as well, and use non-chemical control methods. So don't plant barley on barley, and reduce your stubble loads if you can. And then try and spray strategically and rotate and mix your fungicide groups always. So further research trials into investigating control measures of these triple mutant net form net blotch isolates will be ongoing, so that we hope that we can provide much more information about this in the future. If you would like to visit our website, we're at afren.com.au. We have a whole host of resources that you can discover there. and hopefully get you some more information that you require. So thank you for that. If anybody has any questions in the chat, please feel free to put them into the Q&A section down the bottom. We'll see if we can do that for you. I'll give a few minutes for people to ask some questions if you have some. Well, it doesn't seem like there's very many questions. I'd like to thank you all for joining us on behalf of Lisle and myself. We would really appreciate your feedback. So there is a link in the chat section, and that would be really good if you could and do that survey for us and give us some feedback. Thank you very much for joining us today. Sorry, I have just received a question. Why do we still keep these varieties that are so susceptible? Well, I'm not sure I can answer that one. Lisle, do you want to take that one on? I would imagine it's because you get returns on those particular varieties.
Yeah.
Is that correct, Lisle?
Yeah, a lot of the susceptible varieties are high yielding, and yeah, for that reason, they are still quite regarded as important in the system. However, I've seen a lot of the varieties kind of not being used as much anymore because yeah, they just become too hard to manage.
Yeah. So I suppose it just takes a bit of a culture shift away from those onto something that's a little bit more resistant. Excellent. Another question we have here, do the resistant varieties resist against other diseases or pests? Do you want to take that one, Lisle?
Yeah, I can. The best is for variety choices to look at the NVT ratings. Unfortunately, most of the varieties, they lack in resistance for one or more of the diseases. But there are some varieties out there that are not necessarily sitting at that really susceptible end of the scale, but they have some level of resistance. So the best is, if you want to pick a variety for your specific environment, look at the diseases that's of concern there, and then look at varieties with some level of resistance to those specific diseases.
And I suppose where you don't have that variety resistance, then you should also be looking at your other integrated disease management practices to make sure that you're using those in conjunction with resistant varieties or not so resistant varieties.
Yes.
Oh, hang on, sorry, there's another one. I'll do one more because we have time. What fungicide actives or strategy would you recommend for net form net blotch as the main disease? So AFREN would always recommend that you rotate your fungicides. So you need to be using different fungicides each time you spray. So not only different fungicide actives within the group 3s especially, you want to be rotating the group 3 actives as well as rotating each of the fungicide groups as well. Yeah, that would be our best recommendation, I would say.
And maybe just recommend, or just add to that often these diseases, they can last quite long into the season. So also remember to look at your withholding periods of the fungicides that you do use at the back end of the season, and then also look at the number of sprays that you are allowed in a season, and make sure that you don't exceed. That's specific for some of the fungicides, such as tebuconazole.
Great, thanks, Lisle. I think we can definitely call it a day. Thank you very much for joining our AFREN webinar today. We hope that it's been informative for you, and we'd really appreciate it if you could do our survey at the end.